if that's okay with all my co-presenters. So can I share my screen at this point? Okay.
And you're in charge of introducing yourselves too, and all
We're
your
all going to
presenters.
introduce,
How's
yeah,
that?
each of us. Okay, we're just gonna co-presenting the whole thing, 3 of us. All right, can you all see my screen?
Yes.
Okay,
Yes.
I'm gonna start the slideshow. All righty.
Ooh, I like that.
So I want to say something about myself first. I'm Daisy Zhang, and I'm from Del Mar College, Corpus Christi, Texas. And I started my work at Del Mar College since 2010. And I've been teaching all the biotechnology courses, all 7 of them, for the Department of Natural Sciences. And Rob, you want to introduce yourself?
Yeah, hi, I'm Rob Hatherill, and I've been a professor at Del Mar for about 15 years. And so I basically teach majors biology, microbiology, things like that. And Daisy and I have been able to really collaborate between, the biotech kind of field, and we use my class as kind of a seedbed for recruitment. This is our chair, Jack Southard. I'll just mention he's a great guy. He supports what we're doing. And then go ahead, Susan.
Okay, I'm Susan Woodard, and I am at the NCTM at Texas A&M University. We're a workforce training center, and we started working with doing a training for Blaine College back in 2016 or 17. And after that, I decided that community college partners was the way to go, getting our training to as many of them as possible. And we partnered, I met Daisy and Rob, and we were successful in getting this training, some funding to support our training finally. So I'm looking forward to working with everyone.
Thanks, Susan. So a little bit background story of how this all started. So it's about 3 years ago, Rob and I took a long road trip to visit the center, the experiment station. And we visited the lab, and it's all industry standard, very impressive. We met a lot of personnel over there, and on the way back, and Rob and I were just talking like, how are we gonna, collaborating with Susan? And then I remember, by involving AC2, Linnea kept talking about biomanufacturing certificate. So that's like ding, that's how the whole thing started. We submitted the grant, I think the first time it was 2018, right, Rob?
Yes.
Okay, 2018, we didn't get very lucky. And then we tried again in 2019, and thanks to Susan, we got more industry letter, and we got funded in May. So, and then in the middle of pandemic, of course, we hadn't been able to do anything, to be honest. So involved in this workshop, I feel like I'm just for me and Rob, at least I feel like this, I'm totally a newbie in this whole society. So we're here to more like learning how to start this whole thing than presenting what we have done. This is how I feel, at least. You want to add something, Rob or Susan?
Go ahead, Susan, if you have something.
I guess I should probably point out that the first person I met was actually Carol Twitchell at Collin College.
Right.
But so far we haven't launched anything yet, but we can.
Carol is here, by the way. All right.
Yeah, Carol's on the call. And so is Bridget.
Uh-huh.
Okay, I'm gonna move on. All right, so I want to do some introduction about our institution. So Del Mar College has been founded in 1935, and it is a Hispanic serving public community college. So we have pretty much 70% or higher of students are Hispanic, and over 70% are female. So talk about underrepresented students, Del Mar College probably like on top right there. And the good things about the biotech program and our upcoming biomanufacturing program is the bond has supported to build a brand new Southside campus. So I think it is this building right there is going to be dedicated to as the STEM building. And Rob and I will get moved to there when the building's finished. We will have, between me and him, I think we'll have almost 5 labs, right, Rob?
Yes.
And a lot of space. And also, I think the school is going to support us with brand new equipment. So it's just, it's very dreamy, just the whole situation. So this cannot be a better time for us to start another program, which is the biomanufacturing certificate. And the reason we all got moved there is after Rob and Zhang started a program, biotechnology program in 2008, and I joined 2010. After 12 years of effort from all of us, and has become, according to the school, one of the major signature program for the Del Mar College. So we got very lucky. We're going to do the whole program, not just biomanufacturing, the 2 years associate or 1 year certificate, everything to the south side, brand new building. So that's absolutely provide infrastructure for upcoming, whether it is undergraduate research activity we're going to do, or the biomanufacturing certificate and all involved the lab, brand new equipment, that's make it possible. Anything else you want to add, Rob?
Yeah, can you see my cursor, everyone, when I move this or not?
No, I have to make you throw this to you if you want to share your screen.
Oh, okay.
Where do you want me to point?
Well, just point to the top floor, because we have the top floor of this building
Right
in the
here?
middle.
Right here?
Yeah.
Okay.
And yeah, it's a great opportunity. These are, like Daisy mentioned, the signature programs, the centers of excellence. And so we're the only one from STEM in our department, natural sciences, that's moving over here, and has office space. We'll probably, well, any event, and it doesn't look like this right now, by the way. This is an artist rendition. This is all still under construction. It probably won't be ready for, I don't know, a year and a half. So this is just all weeds in here right now, and construction trailers. So this is this is kind of a little misleading, I guess. I just wanted to say.
It will look like that, I promise.
Yeah.
So Rob, do we have this whole floor? Do you know if we have this whole floor? I think we do, right? This whole thing.
Yeah,
right there.
well, science, there will be part-time people for chemistry. There'll be chemistry labs as well. But yeah, it's really focused. Daisy will have a dedicated cell culture lab and the whole 9 yards. So it's going to be pretty nice. I even ordered, since we're on the 3rd floor and the roof, at the time of construction, I decided to order biosafety level 3 cabinets for the labs, because it doesn't take that much to put a vent on this ceiling at the time of construction. So we'll probably never go BSL 3, but we have that capability if we did want to. But I don't really want to go BSL 3 anyways. We routinely go BSL 2 in our labs, but 3 is a little maybe special projects or something, we would do that. But in any event, I just want to mention that our students, a lot of them come from, I hate to say it, but a lot of them in South Texas come from poverty. And this is what we've done. I kind of give you a little background is Daisy and I have a lot of nighttime student poster events, and we really, really strongly encourage the families to come to those, because then they basically see the importance of what they're doing. And we've had a lot of successes along that line where students, we have a couple that are at MD Anderson and University of Houston, and they're collectively making more than both of their families now. And so they took their first vacation. So those things kind of drive us. Anyways, go ahead with the next slide, Daisy.
All right. So I also want to do some introduction about our program. So currently what we have is a 1 year certificate and a 2 year associate degree. And our class size is about between 10 to 16, so it's very manageable. I mentioned yesterday, I'm even teaching hybrid class right now. This semester is finishing up right now. I have about 10 students. With everybody with the proper PPE, is doable to teach biotech in lab for sure. And so majority of our students, they're freshmen with 2 or 3, maybe TMCC graduate, couldn't find a job, and they would join us for a 1-year certificate. Immediately after internship, they got hired. And we do have 2 on-site, on-campus embedded high school. One is the Howard branch, the other one is the Collegiate High School. Currently, we have articulations with both high school. So what those high school students are doing is they join the high school and also start a 1-year certificate. And since our grant is funded, we're going to actually start from next semester to work on this curriculum to pass all the paperwork for our degree plan curriculum committee. We have all the intention to get that articulated with our 2 on-site high school as well. And we currently right now produce about 8 to 12 students per year. That's including the certificate and the associate degree. And I know it's not a lot of people like Austin Community College, but consider there's just, 1 faculty, which is me teaching biotech class. So I can't, we cannot get more graduates than, this number right now. So articulation agreement, we have already articulated with 2 schools. And although I don't know what's going to happen to Texaco, I haven't heard from them ever once there was a personnel change. But most of our students actually, if they do not go ahead work in industry, they like to transfer to the TMCC. And later, I think near the end of today, you will see a student of us, Daniel. I don't know if he's already here or not, but Daniel was one of the students who transferred to TMCC. And then thanks to Susan, Daniel is working in Austin right now. So we'll see him presenting as I think the last session of today's workshop.
So Daisy, he's right here in this picture, with the poster. He won, first time he went to a scientific meeting, he won an award.
Oh, right here. Yes.
Yeah. And he's, I don't know if he's smiling because of the award or all these ladies are draped on him, but basically.
And he's here on the call today.
Yeah, it's in the last session. Yeah.
But anyways, this is a big group we bring to a regional ASM meeting. And I don't want to brag, but we swept the awards. We took 1st, 2nd, and 3rd place in the phage biology. And then we also took, there's only 1 award for the undergraduate presentation. We took that. So we kind of, I guess, made a name with our program pretty quick going to these regional meetings because the students were, just so great. They just really were competitive.
All right.
Oh, and then point to the bottom right. So this is Molly Robertson, and she's got a great career track job. And she is one of the students that I took to Berkeley Lab. I was a faculty fellow there for 12 years. And this is her PI there at the lab. And that also was a conduit for a lot of student successes because they were, it's hard to replicate a national lab, at your college. I mean, it's almost impossible, the kinds of equipment that they have at the national lab, especially the Berkeley National Lab. And I know Jim DeClos was out there at J-Bay. I saw him in a meeting, but we didn't get to talk. So Jim, you're also involved with the national lab. Okay, I guess he's not there right now, maybe.
Are you done, Rob?
Yeah, done. Go ahead.
Okay. Susan, are you ready? This is your slide.
Oh, sure. Okay, so the way we designed our part of the program was that once the students were, I guess, had some experience doing research. Oh, I guess I should put myself on, sorry. Okay, once the students had some research experience in the labs at Del Mar, they would be ready to take some online training courses. And we make these courses available to anybody who comes and does training at NCTM. There's 5 different modules, starting with introduction to biopharmaceutical manufacturing. Well, actually pharmaceutical manufacturing. And we describe the difference between pharmaceuticals and biopharmaceuticals, and cover a little bit on vaccines as well. They then take understanding the science of the biopharmaceutical industry. They take CGMP procedures and documentation, which I think is a really good one for people wanting to get into the industry. They then take pharmaceutical facility operations. That's one that I review regularly, just so that I remember all the nuances of what it's like to really work in a clean room and all that fun stuff. And then finally, safety in the biopharmaceutical industry. And they have some online assessments. They're somewhat interactive courses with assessments as you go. And once they complete those, they're ready to come and take things on site with us. And it's a fairly accelerated training that we do. And we count on the information that they get in that online training to allow us to be able to hit the ground running. They get a lot of the jargon, familiarized with the jargon of the industry. And hopefully when we say things like CFR 21 Part 11 compliance, they know that that means electronic data systems and things like that. So anyway, so then they come on and they do this 2-week hands-on training where we essentially do kind of what you guys have been talking about, but we do it on a slightly larger scale. We don't actually transform the bacteria ourselves. We have stock of our GFP already in E. coli. We take and we make media according to SOPs. We then inoculate or start a seed culture. We then inoculate our bioreactors after we make the media and sterilize it in place, grow the cells, monitor the operation, very much like what Maggie was talking about, doing some of the assays, following SOPs, and then harvest the bacteria, and then we start our downstream processes where we actually purify it. We have some non-GFP related stuff that we do as well in the downstream, and we try to intersperse elements of quality throughout the training. We used to have a 3-week training where we did quality up front, followed by upstream and downstream, but to save money and time, because it's really impractical to be here for 3 weeks, we have this condensed training now that we do, where we emphasize upstream and downstream with a little bit of quality activities mixed in. So, and then the unique part of what we want to do with the community college students is to prepare them for interviews, because I think that's where there's some hindrance to being able to get a job, because you just don't know. We would want to review their resumes and then go through some mock interviews with them before we then put them in touch with some industry partners who might want to be first in line to look at them for jobs. And we have a number of companies that have agreed to interview trainees coming out of our program. So, any questions on that before I move on to the next slide?
Can I chip in some information? So when we got the grant funding, the grant also going to support me to go with 3 cohort of students to join your training. So one of the main purpose to do that is I was not trained as a biomanufacturing specialist. My PhD was in plant molecular biology, and then I learned all the cell culture stuff when I took Delmar's position. So this is new for me. So by doing this kind of also faculty development, the idea is, I think Rob probably going to go to 1 year as well with me. And we don't know if I have the funding or not, but the grant support one person. The idea is after the 3 year after grant finish, I could be able to do those trainings similar to what the center can offer in our brand new building. That's what I'm most excited about. And let's see the next slide. So go
Yeah,
ahead.
thank you, Daisy. That's a good point, because there are things that we do in our trainings that you'll be able to try and incorporate in your teachings.
Right, yeah. So you're not just training our students, you're training me. Mostly, actually, probably the most important person to get trained is me. Yeah.
Well, you
Great.
also have online version of it too, right, Susan?
We have online, we don't have online hands-on anything. We are, of course, thanks to COVID-19, talking about that. And we are trying to outfit a space where we can do our lectures remotely, so that if we have to, we can do some things. Like we could do gowning training remotely. I don't know how we would, if we would send you materials so your students could do it, but we absolutely could walk you through gowning or something like that. So, yeah. Okay.
This is also your slides, Susan.
Okay. So I guess we were asked at one point just what were the other things that we were working on. This was in preparation for the big ATE meeting. And this is the slide that I made about what we have going on in our labs now. I can't say enough good things about NIMBLE. Its membership in NIMBLE is for community colleges a bargain. Texas A&M invested in this very early on, and we're very happy that they did, because we've really been able to get a lot of interesting projects funded through NIMBLE. They are not completely funded because we have to come up with cost share. And membership is expensive for a university that's a large like Texas A&M. But we've had, I think we're on our third project now. But the one we're working on currently is a gene therapy training network project that was funded to North Carolina State. They have a gene therapy training that they developed with help of some industry partners in their area. And it's really been interesting. We are going to make AAV2 particles using baculovirus in insect cell culture. So it's how a lot of people are doing it. We actually are doing it. We're doing the feasibility on a small scale right now before we scale up to 25 or 50 liters. But the reason it's a really neat platform is because it's BSL-1. So we can train easily without having to get approval to train people on a BSL-2 process, like making it in HEK293 cells. Instead of that, we're using insect cells. So it's a really neat process. And we are also, through NIMBLE with funding from NIST, we have some money from the CARES Act to make spike proteins that are being used by different researchers for screening. In the early days, it was for screening convalescent serum plasma for protective antibodies, and then to screen antibodies that those researchers are making after isolating the genes from the B cells to identify, after they've identified protective antibodies. So we regularly make, we spent our summer making SARS-CoV-2 spike proteins in different forms and supplying those to people. And that was really a great experience for us because it kept us current in all of our, we don't just teach, we are able to do, and we learn a lot when we do that. Then we also pretty much can't do anything else in our labs right now because we are regularly training cohorts of 10 people that are mostly new hires at Fujifilm Diosynth Biotechnologies. They've been hired to support COVID-19 vaccine manufacturing. The facilities at College Station will be dedicated to insect cell culture made vaccines. So the first one will be Novavax's vaccine. And that should be starting up very soon. It's exciting. So we're spending a lot of time putting people through that training, just a rudimentary training, because they need a lot of employees to support that work. And we're trying to do it with social distancing, which is really interesting. and then we also regularly teach students in the professional masters of biotechnology program. We do a lab for them, and that lab is very much like uses some of those same activities that you guys use in community colleges. In a lot of cases, we just take it up a notch, where maybe we have them calculate for the plasmid that they isolate and the number of cells that it came from. Is it a low copy number or a high copy number plasmid? So it's very similar to what you guys do, but we have those activities just be a little bit more advanced and involve some calculations. And then we're always trying to expand our curriculum into new areas as things change in the industry.
Susan, I do have two things that I want to talk with you. I think it's interesting, to bring it up in front of the audience. The first thing, the first thing is you're still offering that a 2-week certificate to all the people who went through the training with you, right? That will include Delmar students later, right?
Yes. Yes.
Okay. And then the second thing is, well, based on our last seminar from yesterday, I look at how educators can get the timeline for us to get vaccinated, which can be early as April, can be late as middle of August. So I think, if Delmar lifted up the travel ban on the students, allow us to bring students, let's say next summer, do you think it's gonna be a complication from your side to housing those students and have them in your lab?
I have no idea. I would think the university would be happy to have them, but I don't know. Texas A&M's never really shut down, so we've done a lot of interesting things to accommodate staying open, and I think
Well, I guess we just have to wait and see, right? Nothing, we can't plan for this for the first summer yet. We have to wait.
Right.
All right.
But there's the beginning of the summer, there's the end of the summer.
Yeah, right.
Things could change a lot.
Right.
Okay, so Susan, can you comment on that 2-week intensive training program? Because I know you surveyed about 75 content experts, and it's really well accepted. Like, was it Lonza that now has all of their employees go through your training program? Forget the company that you mentioned.
Oh, no, it's not Lonza. So we license our training to at least one company, and it was made public. That's Cytovance in Oklahoma City. So yeah, but they license a subset of activities that they handpicked, and then they tailor it to their needs. Is that what
right? You have a whole database of different modules, so you can kind of customize it. And can you just give an overview of, because I want to give you a plug, because I think it's a great program. You have 2-week intensive program. I think you start with upstream processes first, and then the second week is downstream. Is that right?
Yes, that's the way we currently do it. Although I think we do quality activities on the first day of each of those. And that doesn't mean that's exactly what we would do with you guys. We proposed something, and honestly, I didn't remember what we proposed. So we can look at that again and make sure it's still a good fit.
Okay.
But originally it was, we did military veterans training, and we started with a week of quality, and some, what I'm just going to call remedial training, where we do pipetting and weighing and pH and conductivity measurements, just to make sure everybody knows how to do that. And we'd probably do that for these students as well. And then, so we had this quality week, then an upstream week, and then a downstream week. But we are doing it in a more condensed format now.
Okay.
Rob, take it over.
Okay, so this just kind of gives you a quick overview of the goals of our project. And obviously, we're trying to get this new advanced level 2 biomanufacturing certificate. And basically the same thing. We have a customized workforce of underrepresented students. And one of the reasons that really motivated us is especially the Houston job market. I know Susan mentioned that I think it was one of the companies, I'm not going to mention any names, but one company had sandwich boards, and they're just trying to get all these people, and they were because they just can't fill positions at their company, and they're trying to grow. And so we're targeting the greater Houston area for our graduates eventually. So in any event, that's kind of the real, I mean, it's really the underlying goal of the project is to provide a pipeline of, basically jobs for our students, and they can go, hopefully right into after, cause I think this, especially a 2-week program with Daisy's work, I think they can get a job probably with pretty quickly because there's such a demand. What do you think?
Susan, you mentioned, I think from a month ago, the conversation when we were preparing for the ATE presentation, you mentioned that back then you couldn't produce enough graduate from your program for
we get inquiries all the time about that people are hiring for this, that, or the other position, and do we know of anybody?
Right. And I think the pandemics make it even more demanding. Even I can foresee after the pandemic, they're probably still going to be like, some speaker yesterday, they mentioned they made the same statement. So basically Del Mar College we want to cut into that piece of cake as soon as possible. That's why we're working with you.
So the bioeconomy is very strong, and I don't think that that's going to change.
Well, Houston is second in the nation in growth for bioeconomy, and Austin is third in the nation. Of course, California and in the Northeast are always greater, but just the fact that we have greater growth right now, accelerated growth, is good. And I know that for our biomanufacturing program, we do place our students, and we're getting a lot of calls for jobs. So this is the time that we all should get together and work on those skill standards for the state and get them back instituted in the state.
Right.
All right, Rob.
Okay, so then these are some of the strategies for our program, basically, the ATE grant. We wanted to create a certificate, I know that for just an associate, a certificate, it may not have a research component, troubleshooting component. A lot of the students, if they just get a quick degree, 1-year degree, they're going to be just following SOPs. But we do have embedded in a lot of our classes a research component, so they could have a lot of problem solving abilities, which could let them advance quicker at the company. I know that most of them are just going to be following SOPs to the T, because if they deviate from that SOP, they have to document it, and all that. But if they have a problem with the process, then they will have some troubleshooting skills already. So that'll give them a better base for thinking. And we do have a number of industry internships that we're supporting on the grant.
I think it's 4 per year. The grant support 4 industry.
And we find, okay, I'm sure if you guys and gals out there, if you've been using internships, you find that that's really an incredible transformation of the students. I mean, they see, female students see females in leadership roles at these companies, and they get a role model. There's just so many things that, that the internship experience provides them. And it's really hard to assess, because you see them on fire when they come back, and they know what they want to do with, they have a career direction, most of them, and they're kind of on fire by themselves. So anyways, That's about it for that slide. And then we've already kind of talked about this, that Linnea gave a great survey, so I don't think we need to spend a lot of time on that, but we don't have locally here a lot of biomanufacturing, but keep in mind that a lot of the oil folks are getting laid off because no one's driving their cars, right? And so a lot of them are being retrained into biomanufacturing, so that, I mean, they have engineering background. They can kind of transition into that field pretty easily. So once this, you know, who knows when COVID is going to be over? It's probably always going to be with us, right? But once Daisy gets this program going, I think, because we have a fair amount of people that are in the oil industry that are probably laid off here locally, that we can tap right into that, retrain them, and they can get career track positions in the greater Houston area. So that's something that we're really motivated to do, especially Daisy.
Yes, get them hired. All right, so you have one more, Rob.
All right, well, this is kind of redundant, but I don't spend a lot of time on this, but we're also planning to disseminate the project materials through the big center grant, you know, ATE Bio. Am I saying it right? Or I'm probably not. But anyways, and basically get this advanced level 2 biomanufacturing curriculum together and have it approved by our curriculum committee and the state and all that stuff that has to go along with it.
Well, I'm going to chip in. I think the ATE Bio has been extremely helpful.
Yeah,
even it's just been less than a year. For example, there's a couple of virtual, I involved in giving one of those, but I found some of the biomanufacturing virtual teacher resources is so informative, educational. I use it with my students. They're not, well, we haven't had a program start yet, but I have students in Introduction Biotech too. Did the online, I think it's a downstream purification, changing the column, look at the yield of the protein and whether your employer is going to be happy or not. I think you guys know what I'm talking about, that particular virtual lab. They have so much fun doing that one. I personally found it so helpful. Someone want to make a comments here? Yeah, so I'm sorry.
Daisy, I know some people had some questions in chat too about the insect cell line. Barbara, okay. Did you guys want to ask Susan about the availability of this?
Yeah, go ahead.
Yeah, we've been working on, we have a NIMBLE grant that Maggie's heading up, and a few different colleges around the country are involved in, and we're looking at kind of gene therapy and cell-based therapies. Mm-hmm. So I know Maggie also made the comment asking if yes. This curriculum would be available.
I think so. So the contact for this would be Gary Galiski at BTech, which is at North Carolina State. It involves licensing from the University of Florida, the whole, I guess, the BACMID and the recombinant insect cells. And so, yeah, I think it's going to be public. I mean, I think that's going to be made available. I think anything NIMBLE does is made available. But honestly, we're kind of the partners on it, and they're the lead. So it would be the people at BTech that would be able to tell you. Maybe John Balchunas, who's at NIMBLE, would be a good person to approach as well.
Great. We know John Balchunas quite well from our NIMBLE interactions, and just the fact that it's a BSL-1 cell line is very attractive, as you mentioned. Yeah. Just having that model, I believe it's AAV GFP, and using insect cells. And I looked up some of the original publications from University of Florida, and it'd be great for us to make a module like that for everybody to use, to get that curriculum available for everyone.
Yeah,
So,
and a lot of companies are using that format now. So
so it's very relevant.
it's relevant. Yeah.
Thank you.
Sorry.
So
I'm looking at the chat very much.
no, that's all right. It's hard to do both at the same time. It's too bad we're not in a living room staring at each other. It'd be easier, but well, just let us know what we can do to help. And remember that Maggie's head of the regional center grant. I still say you're head of the regional center grant, so and you will continue forever.
Well, I am still. So
Yes.
funding is still ongoing for a little while longer. So I'm happy to
Yeah.
answer anybody's questions about NBC. Absolutely.
Yeah. Yeah. But
Hey, folks, Lenny, I have a really quick question for everyone. I think that might be useful for a lot of folks that are thinking about insect cell lines. For introducing this into the laboratory and node programs, do any of you guys have a breakdown of cost for bringing these things in? Because we had looked at it before, and we were looking at insect serum and qualified insect serum, and the cost for bringing that in is a little bit high in comparison to the other serums. But if one has a breakdown of cost and how we can actually integrate that into programming, that would be super, super helpful, I think, for the community.
I agree, Tom. The one thing is, I know we did insect before, and I learned it at Penn State. We did it because you could grow them up at room temperature, and you could grow them up in spinner flasks. And they also didn't require CO2, so they're a little bit less equipment intensive. Now, I don't know about these guys. Susan, you have to tell me about that. So some of it outweighs itself in terms of these things, even if their serum is more expensive.
Yeah.
And these were light sensitive too, so.
I can't speak to the cost because I'm on downstream and assays. So I do think it's not trivial. And then we're going to be using a rocking bag bioreactor. So you have the cost of the bags, but that's because we're going to do a larger scale in our training. So.
So for the training, Tom, for cell culture, I just brought a set of just one cell line for insect, and from ATCC, along with I think the 1 liter media to go with it. The media itself is really not that bad, and then also got they have a safe version of coronavirus. So I'm thinking about doing some kind of experiment next semester for cell culture. The whole thing come down under. We're talking about the virus, disinfected virus, the insect cell line and the media for the classroom teach. You can get it under $700. Wow. So if you just think about teaching students, I don't think it's that pricey. Maybe I'm spoiled by my budget or something.
So we run a course with about 16 to 20 students in our cell culture class. And I looked into the possibility of bringing SF9s and Schneider snails in because of what Linnea was saying, that you can grow them on a bench or in the absence of CO2 incubators. And when we started to cost it out, it wasn't for the consumables or the cell lines. It really filtered down to the cost of the media, which was, I think at the time, for 100 mils, it was $200 or something like that. And at first, something like that. And it was just something that was just absolutely, we couldn't do it at scale up for our courses. And so we kind of put it to the side. But I would love to see what you have, Daisy.
I'll send you the confirmation I brought for my next semester cell culture. I think it's interesting. I mean, it's not that pricey right now from ATCC.
Great.
Well, not to give everybody heart attacks who's on my grant, but I know that through my bio organization, which is THBI in Texas, I can leverage them to possibly get things cheaper. And then we've always talked about the idea of leveraging the center grant for mass purchasing to get things cheaper. At some point, guys, I think we should explore this. I probably just made everybody at my leadership team faint when I bring up some new thing like that. But, there's always power in the many.
So can we go back to the barrier of our current topic? Just to finish up this presentation.
Sorry.
Is everyone is okay? So we've been talking about, we already mentioned some of the major things right now, no bigger barrier than the pandemic right now. So we already, Rob, you mentioned that several times, we had to postpone the grant for maybe 1 or even 2 more years, right?
Right.
So, and then we have to travel. There's no any other way we can get this done without have to students for the first 3 years, bring students travel between DMC to a college station, which, you know, this right now what we're dealing with is really complicate the program. But, you know, cross the finger, we'll get out of this sooner than later, I hope. And Susan, do you want to bring up any barrier that you think might happen once we start working on this?
I don't know if it's once we start working, but just to prepare for it, we keep running into supply chain issues regularly. So we can't get some of the gowning supplies. We have quite a stockpile, but we recently ran into issues with 200 microliter pipette tips. You know, I did not see that coming. But just hollow fiber cartridges that we use in our tangential flow filtration activity just take months to come instead of in a few weeks. So anything we need, we have to order very much in advance. And at the same time, you know, be aware that we are actually competing for some of the same consumables that companies are using. So we don't want to take gowning supplies away from companies that need them right now. So
How about -70 freezers?
What's that?
Okay.
-70 freezers. Try to get one of those.
Yeah, good. Rob, do you want to add anything on the barriers before I move to the degree plan?
No, I think you guys covered it pretty well. Thanks.
Okay, so I haven't really have this degree plan passed. This is what we have put in the grant. So it's very overlapping with our current degree plan, just general 1-year certificate of biotechnology. So 2 things we is never has been offered in Del Mar College is the we're going to modify our internship course, which was a capstone course, and then to maybe more towards the biomanufacturing. I'm hoping we can actually incorporate the NCTM, those 2 weeks training into this one. But right now, this is not the final one. I'm debating whether to have it in the summer internship or in the summer one, we actually put in the special topic in biological technology or technician, this intro class. So those were the 2 things that is in progress to be determined. Again, Susan mentioned everything is kind of has to be worked out in detail. We just started. So, but most of the biotech classes, including the biology 1, 2, will be taught by me. And then the biology 1406, and I don't think it's a micro in there. No, I think most of the biotech students, when they're taking the biology concept, I will be taking from Rob. So that's our current degree plan, but I think this is not this might not be the final one. It's also,
You know,
I'm sorry.
it's perfect, Daisy, because that's what we do right now.
Because we copy you guys.
I'm sorry, I know I interrupted you. I'm going, but now we just need to go to the state as a group. You know that. We do everything in Texas as a group.
I mean, also for skill standard, you know, whenever you want to get that start, we're in. Okay.
Sorry, I'll be quiet now, but I was just excited.
No. This it's supposed to be supposed to be right, informal, right?
Yeah.
All right. So anybody want to comment on the degree plan? I mean, I know there's a lot of pioneers in this Zoom meeting.
Daisy, you had mentioned earlier that you have some good articulation agreements. How many of these courses articulate and transfer? Is it just your BIOLs, or do any of the BITCs transfer?
So yeah, right now with TEMCC on this particular degree plan, so one hasn't happened, which is the introduction to biomanufacturing. This one will be only one, no transfer. Everything else you're looking at here can be transferred to TEMCC. We have a very good deal. Yeah.
I want to correct you, Daisy. I should tell you that last summer, my group can tell you that UT actually articulated with the biomanufacturing.
Really? I did not know that.
Yes. Wow. So we have to bring you all up to speed on that. Wow. So yeah, UT actually took biomanufacturing because we did a cohort of UT students through our program. Okay. And so they took biomanufacturing.
Then on that note, I'm going to push my connection in MCC, Jeff Session. You all know him, right? I'm going to push him to get this course in our next renewed articulation. That has to happen. If UT are taking yours, they have to take ours. But of course,
yeah, and in fact, UT paid for their tuition too. Wow, and they also took the regulatory course that we have.
Wow.
So they took both courses just to get UT students into biomanufacturing.
So is that separate, the regulatory plan from the degree?
Yeah, we actually have it separate. We have an online course that's BITC 13. I know we're just going off on this stuff and the rest of the people who are listening are just going, what in the world? So obviously Texas needs a meeting together, but yeah, UT took both BITC courses.
Daisy, I have a question.
Yeah, sure.
Oh, can you go back on that slide? So I know that I've asked you questions about your internship before, and I know you do a summer internship class where you bring in a lot of students in. Is this 2-week training at NCTM substituting for your internship, or is that an addition to?
I just mentioned that I haven't made up my mind yet because we're coming. Okay, so the internship is required not only for this certificate, it will be required for 2-year associate, right? Mm-hmm. Also required for other just certification that we already have. So I think I'm either going to make it kind of like a tailored to whatever the student major in, or I should one other option is I'll move this 2 weeks NCTM training into the biotech one. So I'll offer this class in summer one if we can have that 2-week training just incorporate into that summer one course 6 week. Mm-hmm. So I will do maybe 2 weeks before the trip, 2 weeks after. It's the detail that we have to work it out. Depends on the schedule also with Susan. So I can answer your question basically at this point.
Usually, how many hours do you do when you do your own course? What you do in the summer for the interns? How many hours do they take?
Right now, we changed the internship from 3 hours to 4 hours now.
Oh no, I don't mean how many credit hours. I mean how many hours do they spend in the lab?
How many hours to spend in the lab? Geez. So if I teach my intern, if my intern doing research with me on campus, I require them to at least spend 10 hours per week.
Okay, so probably 100 hours for the summer then?
100 hours for 10 weeks, yeah.
Wow, our interns have to do 320 hours because I've been thinking, how could I do something like that? Because I'm sure that helps with the graduates. But 32 hours a week that's a lot. Thank you.
Oh, no problem. I don't even know if I answered your question or not. But
Daisy, I have a question about your internship program. Do you place the students or do they have to find their own? And how?
Both. A lot of our students, and Rob can copy me on that. A lot of our students have a lot of our students actually have done a bunch of undergraduate research during the class they're taking with Rob or with me. So we try to help them to build a very impressive CV by the time before the internship. So we write recommendation letters and we fund all those, extra funding internship, encourage them to apply. So almost, I would say at least 1/3, some summer, maybe half of students can get internship themselves. And we just support a little bit, maybe for the course fee, they enrolled into the internship course. And for the students who cannot get extra funding, we have grant to cover them. And Linnea has helped support some from the AC2 grant. And then we have our other NSF-ET grant to support them. And then if those grants run out, we have so many research projects in summer, and I would just take several interns myself to work with them. So we have different options. And there is students who really do not want to leave the city anywhere. but they want to have that research experience. So they might choose to work with me in my lab.
Daisy, you also received funding from the foundation office.
Right. Yes. Thank you for reminding me. Yeah, the school very supportive. They've been funding me altogether almost, I see, over $40,000 for the last 3 summers. So yeah, I'm spoiled in a way on the funding.
I have a question. This is Isa from Massachusetts.
Hi.
I can see you have 2 introduction to biotechnology courses.
Yes.
I'm curious what each of them covers.
So you wanna ask what I teach in both of the classes?
I wanna know why you have 2 intro to biotech courses.
Okay, my intro to biotech 1 focus on all the technology involving DNA. So basically all the way from very basic making, I do spend a lot of time teaching them how to make that biological buffer. So very basic, very good solid lab skill take about 1 month, and then followed by the bacterial transformation, PCR, and then DNA electrophoresis, and maybe sometime I'll do some RNA work. So everything that important, some basic cloning in around the DNA, that's the focus of the Biotech 1. Biotech 2 is all about protein. So we normally in the lab doing I feel like I'm going over the time. Only 2 minutes left. How am I doing on the time, Purnima?
Daisy, you're doing fine. You have time until actually 15 more minutes. Or
okay.
So, yeah, we're good.
Okay. So back to the question. In Biotech 2, we focus on protein work. 70% of work is like hands-on. So we do like a DHFR, that big project from Biorat, DHFR isolation. Is kind of a lab version of biomanufacturing. In a way, we use a lot of affinity column and size exclusive column. And also we do a genomic annotation, bioinformatic. So it's made of a bioinformatic work, maybe 1 or 40% of work, because we're in the HHMI project. In the Intro to Biotech 2, we gather sequence back of bacteriophage. We have to teach students how to annotate the genomic DNA, how to do the gene calling. And then the 60% will be some kind of protein expression purification project that is very close to the biomanufacturing, like a classroom version of it. Does that answer your question?
Yes, thank you.
Okay.
Let me just say that so in Texas, we adopted the Washington Skills Standards. They became Texas Skills Standards. And then all the biotech programs got together and divided them up among core courses, which included biology 1414 and 1415. And so we may have different topics, but we have the same skill standards outcomes for each of our core courses. And Bridget Kilpatrick and the entire group were responsible, especially Bridget, in making sure that biology 1414 and 1415 are core electives so that they transfer to four-year schools to make our dual credit program successful, because they're part of our high school programs too. So we have dual credit with those two. And this mirrors our entry-level certificate to some degree, except this adds in biomanufacturing and cell culture. So in Texas, we work as a team because the state enables us to do things together that way compared to other states.
Daisy, this is Barbara from California. Jim and I have the bachelor's program in biomanufacturing. What I would suggest is quality and regulatory concepts somewhere in your program.
Okay.
Although, genetic engineering and introducing genes into cells is important from a fundamental biology perspective, it's really not something a biomanufacturing technician would actually do. But what they do on a daily basis is work in a regulated environment. So it's really important that you include concepts around quality and regulatory affairs, and even some basic statistics would be helpful.
Yeah, that's great. And that's why I'm here to hear you guys' comments on this.
So Daisy, that's our online course that you don't have.
Yes, please help.
Good, put that in.
It's always good to discover these things in a group meeting that obviously in Texas, we haven't done enough talking together.
Right, yeah. Who do I bother to get that online course, by the way?
Me, and then we can give you the cassette. We gave it to Bridget.
Oh, okay. Maybe Bridget already sent it to me. I'm gonna go back.
But you know, I think what Barbara just said, we should get together and go over the course and think about adding more statistics. So thank you, Barbara.
Yeah, we appreciate that.
The state of Texas thanks you.
All right, so this is probably my last slides, if it's not the second to the last. This is, I mean, like we mentioned before, we're new to this. This is what we will get help from. You know, we already talked about the school support us infrastructure, the equipment, bond money, all of that. And then we're lucky this year, you know, Rob is the PI, I'm the co-PI, the grant got funded from NSF. And, you know, we can start with faculty training, first several cohort students training, and just to get our name out there for those industry in Austin and Houston. So they will give us a chance when we have more students get this certificate. A great help from, you know, in novel ETE bio. I mentioned that just on the online part, how much I have been benefit from using some of the virtual resources. And then Maggie and your team, you know, for the newbies like us, this is like a Bible, I think. Just for example, our dean and our VP, a lady thinking about doing another grant to support us. And then they want equipment list like this. You have to send me right now. There's no way I can put it together. And I went to your book. On that page 398, you have all the major equipment list. I mean, it got me off that, you know, get them off my back just for that email. So we're going to rely a lot on your website, on your manual. Say we want to thank you, take this chance, and your team for coming up with this very, very good manual. And Rob, you want to add on something?
No, I think we're out of time. So, all right, was Cornita there? What's her time? Because it kind of changed, I think.
You have 5 more minutes. No, you're good. You have 5 more minutes.
I need 5 minutes. Rob, you want to say something? We have 5 minutes.
I don't really have much to say. Why don't you go ahead and take these questions? Or I mean, ask the question.
So I think some of the people already answered my first question. I know because we're so new, we're here to ask you question, more like to answer your question, right? So, you know, Barbara mentioned the course and some other, you know, Linnea gave me some information about new course, all of that. But I do have just 2 simple questions. One is for Maggie. When do you think, or if there is going to be another workshop from an NBC2, I'm sorry. Maggie.
Hi, Daisy. I'm coming. We're hopeful for next summer. We have one more workshop to run on the regional center funding, and we'd like that to be a sort of a comprehensive biomanufacturing workshop for people like you, but to be hands-on. So if at all possible, we'll do a hands-on workshop next summer. That's our plan. And, you know, we may put out some feelers to see what people would find most useful. And if you look through our site, I'm happy to discuss with you, you know, anything you find on the website at all that you want to incorporate. How can I help? You know, what type of equipment do we have? Anything like that, really. Equipment list, reagents, anything that will make your process easier and go more smoothly and quicker. We're happy. So don't hesitate to email.
Thank you.
And we're happy to give you anything we have.
Thank you. Okay, we really appreciate that. And I do have a kind of a stupid question because of my own experience. I mean, how do we join Nimble? I tried to join them like 3 semesters ago. My experience was confusing to me. Apparently, there has to be follow-up email or even phone call, but let's just say it's a little bit complicated when we try to join. do you have any, I mean, how do
I can answer that. So if you want to join it, the Nimble is for you. I can definitely say you can do what we did. It has to be an institutional membership, and
right.
your college will fall under the tier 3, I believe, which is $1,000 for the year. So your college will basically, and you can use your funding to pay for it. That's up to your college and you. But you can join in, and if you want, I can go ahead and introduce you to the people who can talk to you about that.
Yeah, please do, because I just when I had contact the supposed to be the person recruiting more membership, and then it's just never follow through. So I probably have to go through you, Panima.
Yeah, no, that should be easy. But remember, the $1,000 membership can't come from federal money. It has to come from state money.
Right.
You can't pay for a grant with a grant, or however that works.
Right.
Or your institution.
Or in your institution.
Mm-hmm.
I think we can put it into our budget, right, Rob? Our biotech budget. We'll just put it on there.
Sure. Yeah.
That would work.
We'll have the campus pay for it. Yeah.
Right. So, all right, I think that's conclude our three-way presentation. And thank you, Susan, for step up without almost no notice at all. Thank you.
Yeah, thank you, Susan.
So don't forget to join Biomade when you can. Everybody.
Okay, Biomade.
Okay.
Writing it down.
We'll have to pull up a little bit of you can actually Google Biomade and find out something about it. So, because that will be free membership, and that'll be synthetic biology.
All right.